My channel, and we are discussing the chapter 2 of the revision notes of MRCUG part 1, and this is embryology, and this is the part 2 of the chapter embryology. Let's continue our topic with our brand new gastrointestinal system. And what are the general facts? The endoderm, they forms the foreguard, the midguard in its continuity with the remaining yolk sac, and vitellointestinal duct, they connects the yolk sac and the midgut. And endoderm also forms the hindgut, liver and pancreases. And splanchnopleurotic lateral phase mesoderm contributes to the connective tissue, the smooth muscles and the serosal layer of the gut tube. The dorsal mesentery, it suspend the gut tubes in the body of the embryo, and formed by the serosal membrane and attaches to the posterior wall of the embryo and extends from the lower esophagus to the cloaca. With the further growth, the dorsal mesentery is lost for the some part of the gut tube, and from the duodenum, ascending colon, descending colon, the dorsal mesogastrium is the dorsal mesentery of the stomach and becomes the greater omentum. The ventral mesentery, they forms at the caudal part of the foregut. It is derived from the septum transversum and covers the stomach, terminal esophagus, initial portion of the duodenum. The liver that develops within the ventral mesentery and divides into two parts: the falciform ligament, which connects the liver to the ventral wall of the embryo, and the lesser omentum that lies between the stomach and the liver. And the mesenteries, they are double layered, allowing the structures such as nerves and the blood vessels to lie within them. A structure is the intraperitoneal when it completely invested in the splanchnopleurotic mesoderm all around. A structure is retroperitoneal when it is not suspended by the dorsal mesentery. Foregut, it consists of the esophagus, stomach, duodenum, pancreas, and liver. The foregut is divided into the cranial portion that forms the pharynx associated with the pharyngeal arches, and caudal portion, and esophagus is the part of the caudal foregut and lies in the posterior mediastinum and give rise to the respiratory diverticulum, and lengthens due to the growth of the thoracic organ. The stomach undergoes a clockwise 90-degree rotation, such that rotates to the right, and this pulls the dorsal mesentery over the left side, and consequently a small gap is formed between the stomach and the dorsal wall called the lesser sac, and the spleen that develops into in the dorsal mesogastrium. And gastrosplanchnic ligaments is the portions of the dorsal mesogastrium that lies between the stomach and the spleen, and the Linorenal ligament is the portions of the dorsal mesogastrium that lies between the spleen and the dorsal wall of the embryo. Pancreas lies in the dorsal wall of the embryo and develops from the endodermal lining of the duodenum. And at least 4 weeks of the embryonic development, 2 pancreatic buds, they arise. The dorsal, they lies within the dorsal mesentery, is the larger of the 2 buds, and its duct is the main pancreatic duct. The ventral, they lies within the ventral mesentery. As the duodenum, they rotates right, the ventral bud, they moves dorsally to lie superior to the dorsal bud, entrapping the superior mesenteric blood vessels between the dorsals and the ventral buds. Both the buds, they fuses, and both pancreatic ducts, they fuse, entering the medial wall of the duodenum. And to form the major duodenal papillae, the tail of the pancreas lies within the double layer of the renal ligament. Lesser sac is limited in its extent by the greater omentum, as the double layer of the greater omentum fuse laterally and communicates with the greater sac by the auditus. The duodenum, it arises the foregut, the midgut. The bile duct, lies in the junctions between the foregut and midgut. The lumen of the duodenum, initially it hollow, and at 2 months of the fetal development it is solid and lie in the posterior abdominal wall, give rise to the pancreas, which lies within the mesoduodenum. Is C-shape, has a dural artery supply. Mesoduodenum is lost with the further development. Consequently, the duodenum is covered with the peritoneum only on its anterior surface, known as the parietal peritoneum. The posterior leaf of the mesoduodenum, they regresses. Thus, the posterior surface of the duodenum is attached to the dorsal wall of the embryo. This seals off the lower extent of the auditus to the lesser sac. And liver is the hepatic diverticulum, arises on the ventral wall of the duodenum and just just above the ventral pancreatic duct, pushes into the septum transversum. Gallbladder arises from a ventral pouching of the hepatic diverticulum. Its duct initially is solid. Failure to recanalize the duct results in the extrahepatic-biliary atresia, and the peritoneum recovers all of the liver except the bare area. This is the area in contact with the diaphragm, and the Kupffer cells. They are the bone marrow derived and the liver line liver sinusoids. At 10 weeks of the embryonic life, liver produce blood cells. Now moving towards the midgut, it starts at the midpoint of the duodenum, terminates approximately 2/3 of the way along the transverse colon. Its artery is the superior mesentery artery, and the midgut they rapidly elongates. But as the peritoneal cavity is small, it herniates out of the abdominal cavity in the umbilical cord. Herniation occurs at 6 weeks and returns at the 10 weeks. This is the physiological umbilical hernia. And as the gut enters the umbilical cord, it rotates 90-degree anti-clockwise. On return to the abdomen, it rotates a further 180 anti-clockwise, resulting in the cecum moving from the subhepatic position to the right iliac fossa. The rotation is about the axis of the vertilo-intestinal duct, and abdominal herniation of the midgut is of the two types. And these are the omphalocele and the gastrochesis. Omphalocele, it indirect herniation and herniates via the umbilicus, covers with a layer of the amnion and associates with the other structures and the chromosomal abnormalities. The gastrochesis, the direct herniation via the abdominal wall, lies in the amniotics and hence is not covered by the amnion and not associated with the chromosomal abnormalities. Then moving towards the midgut forms the second half of the duodenum, ileum, jejunum and ascending colon, proximal to third of the transverse colon. The mesentery of the following organs are reabsorbed: duodenum, ascending colon and descending colon. The transverse mesocolon is the dorsal mesentery of the transverse colon. It fuses with the anterior two layers of the greater omentum, thus appearing that the transverse colon is attached to the posterior surface of the greater omentum. Merkel diverticulum is the remnant of the vertilo-intestinal duct and root of the two, such that it is 2 inches 5 cm long, 2 feet 61 cm from the ileocecal valve, and 2% of the population has it. Contains the gastric mucosa and pancreatic mucosa, and inflammation of it is often mistaken for the appendicitis. Hindgut, they forms the distal 1/3 of the transverse colon, descending colon, rectum, upper half of the anal canal, and forms the epithelial layer of the urinary bladder, urethra. And initially opens in the primitive cloaca, and which communicates with the allantois and the urorectal septum. A band of the mesenchymal tissue divides the primitive cloaca into the urogenital sinus anteriorly and rectum posteriorly. Becomes the perineal body when it reaches the cloacal membrane, and perineal body they drives the perineum into the urogenital triangle and the anal triangle. The cloacal membrane they breaks down at the 7 weeks of the embryonic development, and allantois it is non-functional and connects the primitive cloaca to the umbilicus and becomes the urachus, which eventually become the median umbilical ligament. And in some species, the allantois contributes to the formations of the placenta. Anal canal, the mucosa of the anal canal they derives from the upper half the endoderm and lower half the ectoderm, and the pectinate line they demarcates the two embryological parts of the anal canal, and located at the base of the column marks a change in the blood supply and the nerve supply and the epithelial lining. And anal canal lumen they occluded at the 7 weeks of the by the ectoderm. At 9 weeks this is recanalized. Now moving towards the urinary system, it origins of the urinary system, the bulk of the urogenital system is derived from the intermediate embryonic mesoderm. They forms a nephrogenic cord that underlies the urogenital ridge and develops in the trunk. The craniocaudal development, the development of the urinary system, the pronephrons, the non-functionals and temporary, and mesonephrons are produces the urine, and the metanephrons it produces the urine and becomes a definitive human kidney. The initial structure that develops in the urogenital system Two components are necessary for the development of an excretory system, and the capillary bud and tubules, and mesonephrons they develops in the lower thoracic and the lumen region, and cavities they appears in the mesonephrons, which becomes the tubules and Bowman capsule joined laterally to form the mesonephric ducts, and the mesonephric ducts they drains into the urogenital sinus and forms the bladder trigoon, and in males they produces the ducts deferens and afferent ductules of the testes, and in females they produces the Gardner's ducts. Metanephrosis they consist of the ureteric bud and the metanephric blastema, and the ureteric bud the outgrowth of the mesonephric ducts and eventually grows into the metanephric ducts. They forms the definitive ureters, the renal pelvis, and the calyces and the collecting duct. Metanephric blastema the condensations of the nephrogenic cord tissue around the ureteric bud they forms the nephrons, and formation they continues until 32 weeks of the intrauterine life and functions from the 10 weeks. Bladder they arises from the urogenital sinus after the cloaca they has been divided. Now moving towards the reproductive system. The indifferent gonads they develops from the intermediate mesoderm and is situated on the gonadal ridge, and which is the medial portions of the urogenital ridge. Primordial germ cells migration they starts at the 6 weeks of the embryonic life, originates from the wall of the yolk sac, and cell migrates in the genital ridge and pass is via the dorsal mesentery to the hindgut. It induces the formations of the primitive sex cord, and primitive sex cord is derived from the mesonephrons and overlying the coelomic epithelium. Testes the default is for the gonad to develop into an ovary, and sex determining region of the Y chromosome known as the SRY gene they produces the testes determining factors and promotes the testicular development and induces the differentiation of the gonad mesenchymal cells into the interstitial Leydig cells. Primitive sex cords they penetrates the medulla and form the testicular cords, which anastomosis to form the reed testes. Differentiate into the Sertoli cells and become the seminiferous tubules, which are solid cylinders until after the puberty when they are canalized. And reed testes is continuous with the mesonephric ducts, and the Leydig cells they produces the testosterone from the V gate, and Sertoli cells they produces anti-Mullerian hormone from the V gate. Ovary the absence of the testes determining factors they causes the sex cord in the medulla to degenerate, and formations of the vascular medullary stroma and surface epitheliums they give rise to the second generation cortical cords. At 4 months of the fetal life, primordial germ cells they invest the secondary cords, and the development of the female internal genitalia they occurs in the absence of the testosterone and anti-Mullerian hormones, and initially two pairs of the genital ducts they arises, the mesonephric also known as the Wolffian or the Leydig ducts, and the paramesonephric also known as the Mullerian ducts. Mesonephric ducts lose its urinary functions once the mesonephrons supersedes the mesonephros, and persistent in the males and the regresses in females, and forms the ductus deferens, epididymis, seminal vesicles, and the prostatic utricle and trigoon opens in the urogenital sinus. Differentiation is due to the testosterone being produced from the week 8 of the embryonic life. Paramesonephric ducts persist in the females and regresses in males, and regression is due to the anti-Mullerian hormone and lies lateral to the mesonephric ducts and is a longitudinal invaginations of the coelomic epithelium overlying the urogenital ridge, and the cranial end of the duct they opens into the peritoneal cavity and become the femora of the uterine tubes. The caudal end of the duct they fuses in the midline and regression is due to uterovaginal canal. They forms the uterus, and the upper half of the vagina fuses with the sinovaginal bulb, and swelling on the urogenital sinus and sinovaginal bulb they give rise to the lower half of the vagina. So this is a pictorial development. I'm going to discuss whole of the pictorial development in the third part of the lecture, and you can find it in the playlist. I'm also going to share the mnemonics which can help you to keep memorize this embryology, which is not a very difficult topic but the most asked topic in the MRCG part 1. So moving towards the urogenital sinus, they forms the Bartholin gland and Skin's gland. Paraurethral vestibules is analogous to the male prostate, has the three portions: the vesico-ureteric give rise to the bladder, and the pelvis give rise to the prostate and the phallic. The testicular descent mediates through the guidance of the gubernaculum and is towards the labioscrotal swelling, and gubernaculum in the female they becomes the ovarian ligament and round ligament, and process vaginalysis and outpouching of the peritoneum they from the coelomic cavity protruding into the labioscrotal swelling into the space created by the gubernaculum and precedes the testes into the scrotum, and they forms the inguinal canal as it pushes through the three layers of the abdominal wall, becomes the tunica vaginalis. Has two faces. Independent phase, the testis they reaches deep inguinal ring at the 7 months of the fetal life, and the hormone dependent from the 7 to 9 months of the fetal life. The factors determining the testicular descents: the elongations of the fetal trunk and the increase in intra-abdominal pressures and the regressions of the gubernaculum. External genitalia, they are undifferentiated until the 9 weeks of the fetal life, and the default development is towards the fetal phenotype, and the dihydrotestosterone stimulates the external genital virilization in the male embryo. At 5 weeks of the cloacal folds they forms, and on the either side of the cloacal membrane fuses anteriorly to give rise to the genetical tubercle, and they forms the labia minora and the penile urethra. At the end of the 3 months of the development, the cloacal fold fuses anteriorly, and the lateral to the cloacal folds they are the genital swelling. In the female they form the labia majora. In the male they fuses to form the scrotum, and the genital tubercle elongates under the hormonal influence to form the clitoris in the females and the penis in the male. Reproductive development disorders, the ambiguous genitalia at the birth, the incidence is 1 in 4,000, and etiology is either visualizations of the genetically female infants or the under masculinizations of the genetically male infant. Severity of the ambiguity is classified according to the Prader stage 1 to 5, and the partial or the complete androgen insensitivity. X-linked inheritance disorder is 2 by 3 of the cases. Here this is again a diagram, and now moving towards the etiology of the ambiguous genitalia at the birth: the 46XY under masculinization, the partial androgen insensitivity and complete androgen insensitivity, 5-alpha reductase deficiency, and gonadal dysgenesis. 46XX virilization, the congenital adrenal hyperplasia, and placental aromatase deficiency, and in utero exposes to the maternal androgens. And the oviotesticular disorders of the sexual development, the true hermaphroditism. Prader stages, typical girls. Stage 1 is isolated clitoromegaly. The rest of the genitalia they arises, and internal organs they resembles those of the typical girls. Stage 2, the clitoromegaly and the partial fused labia, and the vagina and the urethra opens into the funnel shaped area. Stage 3, the clitoromegaly and the complete fused labia, the vagina and the urethra they share a single opening, and the urogenital sinus. Stage 4, the micropenis and the labia they are completely joined like a scrotum, and the vagina and the urethra they share a single opening and they need the base of the phallus. Stage 5, the isolated cryptorchidism, and the external genitalia they looks like a typical boy, and the vagina and the urethra shapes share a single opening at the tip of the phallus. Characterized by the normal testicular productions of the androgen. This is regarding the partial complete androgen insensitivity. It is characterized by the normal testicular productions of the androgens and the abnormal androgen receptors, and lead to the incomplete realization of the external genitalia, and the testes they are usually positioned in the inguinal canal bilaterally. Mayer Rokitansky and the Couters and the Hauser syndrome is a congenital malformation of the female internal genitalia due to the Mullerian duct failure that lead to the absence of the rudimentary uterus but with the normal ovarian function. And now let's move towards the next topic. This is the nervous system, And nervous system, they forms mainly from the ectoderm, and formation states at the 3 weeks of the intrauterine life. Neurulation is the formations of the neural tubes day 18 and the neural fold day 20 and the neural tube day 22. Neural plate, it is an ectoderm thickening with the widest at the cranial end, and the neural folds are the lateral to the neutral plates on each side, and they forms a neural groove, and ultimately fuse to form the neural tube and give rise to the neural crest. And the neural crest, they lies beneath the surface ectoderm and the migrates laterally and give rise to the widest range of the structures. And neural tubes, they currently forms the brain, and caudally they forms the spinal cord, and opening at the end, each end of the tubes they call the neurospore. Anterior neurospores, they closes at day 24, and the posterior neurospore, they closes at day 26. They line with the neuroepithelium, and the neural tube lumen becomes the ventricular system. The neural crest derivatives, the brain, the arachnoid and the pyometer, the glial cells and the Schwann cells, nerves, the dorsal root ganglia and the cranial nerve ganglia, and the paravertebral and sympathetic ganglia and parasympathetic ganglia, and the GID. Cells, melanocytes, odontoblasts, and C cells, the thyroid parafollicular cells, also derived from the fifth pharyngeal pouch, and the others, the adrenal medulla and the dermis of the face, skull cap bone. Neural cells, they arises from the neuroepithelium, the neuroectoderm. They give rise to the neuroblast and the glioblast and the astrocytes and the oligodendrocytes and the ependyma and the bone marrow mesoderm. They give rise to the microglial cells. There are four types of the neural tube defects: the spina bifida occulta, unfused vertebral arch, and the spina bifida cystica, the two subtypes. Neural tubes, they are and its covering, they protrude through the vertebral arch. Meningocele, the neural tube, they lies in the normal position, and this is formed by the protruding subarachnoid membrane and the space. Meningomyelocele, the neural tube, they lies ectopically in the cystic space and associated with the hydrocephalus. The rachis, the no neural tubes, and the neural tube is fused with the skin. Spinal cord, they consist of the three zones: the neuroepithelial, the ventricular layer, and the mental zone becomes the gray matter, and the marginal zone, they contains the axons, and entering and leaving the mental zone, and after myelination, this area, they looks white. Has two plates divided by the sulcus limitans, Eller dorsal plate, and the sensory regions, the basal, the ventral plate, and the motor region. And extend the entire length of the vertebrate column until the third month of the intrauterine life, after which it ends at the lumbar vertebral level 2L2 due to the overgrowth of the vertebral column. The brain, three dilatations, they occurs at the cranial end at the of the neural tube: the prosencephalon forebrain, mesencephalon midbrain, and the rhombocephalon hindbrain. Due to the limited space, the tube, they bends at the two places. The cervical, they lies between the rhombocephalon and the spinal cord, and the cephalic, they lies in the mesencephalon. The three primary vesicles, they develop into five secondary vesicles. The prosencephalon, they develops into telencephalon, the bilateral portions, and becomes the cerebral hemispheres. Diencephalon, they becomes the thalamus and the hypothalamus, and the mesencephalon and the rhombocephalon, they develops into the metacephalon and becomes the pons and cerebellum, and the myelencephalon, they becomes the medulla. The ventricular system, the neural tube, the lumen, they becomes the ventricular system. The fourth ventricle, they lies in the rhobencephalon. The third ventricle, they lies in the diencephalon, and the lateral ventricles, they lie in the telencephalon. The cerebral aqueducts, they connects the third and fourth ventricle, and intraventricular foramen, they connects the lateral and the third ventricle. The meninges, the dural sac, they ends at the sacral vertebral level S2, and spinal cord meninges, they arises from the paraxial mesoderm, and the cephalic meninges, they arises from the neural crest and the mesoderm, the choroid plexus, and the dura. Pituitary glands, they develops from the two sources: the down growth from the floor of the diencephalon, forming the posterior pituitary neurohypophysis, and the up growth from the stomodium. The pituitary gland they develops from the two sources: the downgrowth from the floor of the dance phalon, forming the posterior pituitary and neurohypophysis, and the upgrowth from the stomodium, the ectoderm portion of the oral cavity, and known as the Rathke's post, and appears at the third week of the intrauterine life and loses its connections with the oral cavity at the 8 weeks of the intrauterine and forms the anterior pituitary. Now the face and the neck development. The face and the neck, they are derived from a series of the brachial arches that lie on the either side of the stomodium, and the brachial arches they develops in the fourth week of the embryonic life. They arises from the neural crest and the five pairs of the brachial arches 1, 2, 3, 4, and 6, and the fifth pharyngeal arch is rudimentary and disappears, and each arch they consists of a core of the mesenchyme which differentiates into the cartilage muscles and the aortic arch artery, and an inner endoderm give rise to the pharyngeal pouches, and an ectoderm give rise to the pharyngeal cleft, and each pharyngeal arch is supplied by the cranial nerve. The pharyngeal arch is the first is nerve is the trigeminal, and it is the arch of the M, which is like for the mastification, myohyoid, anterior belly of the digastric, and the tensor villi palatine, and the tensor tympani, and the bone and the cartilage includes the malleus incus and the stapes. The second is the facial of the facial expressions, buccinatus, stapedius, and the stylohyoid, and the posterior belly of the digastric, and the bones and cartilage include the stapes and the styloid process and the lesser horn of the hyoid bone. The third is the glossopharyngeal, the stylopharyngeus, and the greater horn, the lower part of the hyoid bone. And the fourth is the vagus. The muscles include the pharyngeal and the cricocorticoid, and the bones and the cartilage include the thyroid cartilage and the cricoid cartilage. The sixth is the vagus, the muscles of the larynx and the esophagus, and the arytenoid cartilage. The pharyngeal clefts, only the first pair of the contributes forms the external acoustic meatus, and the second pharyngeal arch enlarges and grows up a flap covering the remaining clefts and forms the platysma muscles. And the remnants of the lower cleft, they may form the cervical sinus. The pharyngeal pouches they derives from the ectoderms and of the foregut, and forming the laterally as the pockets on the either side of the pharynx. And the third and the fourth pharyngeal pouches, they have the dorsal and the ventral portions. The tympanic membrane is formed from the ectoderm of the first pharyngeal arch and the intermediate layer and endoderm layer of the first pharyngeal pouch. The pharyngeal pouches and their derivatives: the first is the tubotympanic recess and the middle ears and the auditory tube and the tubal tonsils, and the second is the palatine tonsils, and third is the dorsal inferior parathyroid glands and the ventral thymus glands, and the fourth is the dorsal superior parathyroid glands and the ventral the ultimobrachial body, and the fifth is the ultimobrachial body fuses with the lateral lobe of the thyroid gland and the parafollicular cells and they produce the calcitonin. Thyroid glands, they grows from the thyroid diverticulum and divides into the left and the right thyroid lobe, and the thyroglossal cyst, they connects the foramen cecum to the thyroid gland and detaches from the pharyngeal floor and give rise to the pyramidal lobe of the thyroid. The tongue, that develops from the epithelium of the floor of the pharynx, and the muscles invades the tongues in the second month from the occipital myotomes, bringing the hypoglossal nerves and the innervates with it. Anterior two-third of the tongue is formed by the swelling and from the first pharyngeal arch and two lateral lingual ligaments and the tuberculum impar. And the posterior one-third of the tongue is formed by the hypobrachial eminences derived from the third pharyngeal arch and the fourth pharyngeal arch. The sulcus terminalis, the V-shaped group, is represents the line of the fusion between the first and third pharyngeal arches. The foramen cecum is the midline depressions in the circum terminalis and appears as an invagination of the endodermal at the floor of the pharynx and between the first and the second pharyngeal pouches and makes the origin of the thyroid. Now the nerve innervation in anterior two-third, the lingual branch of the mandibular nerve and the chorda tympani nerve, and the posterior one-third is the vagus and the glossopharyngeal, and the valate papillae lies anterior to the sulcus terminalis and the glossopharyngeal nerve due to the forward migration of the posterior one-third of the tongue and mucosa across the sulcus terminalis. Now moving towards the face, they develops from the neural crest of the five swellings, the frontonasal prominences, they form the forehead, nose, philtrum, and the primary palate, and the paired maxillary prominences they forms the cheek maxilla zygoma and the lateral portions of the upper lip and the secondary plate, and the paired mandibular prominences they forms the cheek and the lower lip and the mandible. Event that shape the facial appearance, the nasal placodes and appears on the frontonasal prominences, and the median nasal processes they fuse to form the intermaxillary segments, and the maxillary prominences they fuses with the lateral and the medial nasal process to form the upper lip, and the nasolacrimal groups they separates the lateral nasal process from the maxillary prominences. Ectoderm they floor they forms the nasolacrimal duct, and the upper end they forms the nasolacrimal sac, and the mandibular prominences they fuses laterally with the maxillary prominences to form the mouth and the cheek and nasal cavity. The nasal cavity they protrudes in invaginates to form the nasal pit, and nasal pit becomes the nasal sac, also known as the anterior nares, and the nasal sacs they grows upwards and separated from the oral cavity by the oronasal membrane, and the oronasal membranes they break down at the 7 weeks of the intrauterine life, and the posterior nares they opens into the pharynx, and the nasal septum they develops at the 9 weeks of the intrauterine life, and develops from the medial nasal processes and grows towards the palate, and the maxillary sinus they grows from the lateral nasal cavity and grows into the maxillary bones, and the remainder of the paranasal sinus they develops after the birth, and the palate they develops from the fusions of the primary palate derives from the intermaxillary segments, and the secondary palate they forms by the pairs of the palatine process from the maxillary prominences. Eye development, the placoids they are the thickened area of the ectoderms in the head regions of the embryo, and they forms as a result of the interactions between the neural tubes and the overlying ectoderm, and become columnar and invaginates and migrates deep to the surface ectoderm, and optical vesicles they are the earliest indications of the eye, and the develops at the 4th week of the intrauterine life are an outgrowth from the lateral wall of the forebrain, and induces the development of the lens placoid and become the optic cup. The optic stalk is the connections between the optic vesicles and the forebrain, and the lens vesicles they formed by the lens placoid and detaches from the surface ectoderms and sinks into the optic vesicles. Hyaloid artery the branch of the ophthalmic artery they supplies the lens retina and runs along the choroidal fissures, and the retina they forms the 2 layers of the optic cup, and the outer layer they forms the pigmented layer, inner layer they forms the rods and the cones, and at the rim of the optic cup the retina they give rise to the ciliary body and iris. The mesenchyme they around the optic cup they condense to form the inner choroids and the outer sclera, and the cornea the anterior part of the sclera and the transparents, and the eyelids they develops as the fold of the ectoderm that grows over the cornea, and both the eyelids they fuse initially they separates at the 5 to 7 months of the intrauterine life, and the inner layer of the eyelids ectoderm and becomes the conjunctiva. Ear development is the last topic of this chapter, and the ear developments they forms the 3 different regions: external ear, first pharyngeal cleft, the middle ear the first pharyngeal pouch, and the inner ear the aortic placoid. The aortic placoid they closes the hindbrains and the first part of the ear to develop at the 22 days of the embryonic life, and invaginates at the aortic vesicles. Aortic vesicles they forms the dorsal vestibular portions and the ventral cloacal cochlear portions and give rise to the endolymphatic sac, and the vestibular portion has the 2 parts, the larger utricle and the smaller sacules, and the utricle they give rise to the 3 semicircular ducts. The structures within the aortic vessels they forms the membranous labyrinth. So this concludes our the chapter number 2 embryology, and we are now going to discuss a few related SBAs. We are going to practice them, and also I'm also going to upload the video regarding the mnemonics which will help you to memorize this whole embryology and its development, and also I'm going to share few topics which I will make easy for you to understand, because in this book I have seen that there are the number of the topics which were very difficult to understand. So I'm going to make another separate video for this so that you can understand the whole topic. You can find the videos in the related playlist. So till the next time, take care, bye bye.